Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Kerala SET Biotechnology question paper conducted in the year January 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous years of Kerala SET Biotechnology been answered, you can go to my channel Omni Bioentrance. In the playlist, select for Kerala SET Biotechnology exams, and there you will find. Okay, first question. Nitrogen atoms in urea produced in the urea cycle are contributed by. So nitrogen atoms to urea during urea cycle, they are contributed by ammonia and aspartic acid. Answer is option A. The outcome of the accumulation of acetyl-CoA in mitochondria of the liver. So when acetyl-CoA accumulates in the liver, it is converted to ketone bodies by ketogenesis. It is used as an energy source. A. Acetyl-CoA can enter the citric acid cycle and act as an energy source, but this is not the outcome when they are accumulating. So A is not correct. It has broken down into free fatty acid, again not correct. It gets converted to oxaloacetate, not correct. It forms ketone bodies. So D is the answer. Which of the following are principles of photochemistry? So it is Groth's, Draper and Stark-Einstein law. Answer is A. Raoult's law, Dalton's law, Henry's law, all these are thermodynamic laws. They don't have any relationship with photochemistry. Beer-Lambert law is for spectrophotometry. So answer is option A. Then which of the following has the highest permeability in a resting nerve cell? It is K+. Answer is D. Then which of the following is based on rearrangement of Lineweaver-Burke plot equation by multiplication of concentration of S factor? So Lineweaver-Burke plot equation, if you multiply by concentration of S factor, you get Hans plot equation. Answer is option A. The natural source of bromelain, a cysteine endopeptidase, is. So bromelain, it is obtained from pineapple. Answer is option C. The immobilized enzyme used in the large scale production of high fructose corn syrup is. It is glucose isomerase. Aspartate 4-decarboxylase, it is involved in amino acid metabolism. Hydantoinase, it is in antibiotic production, and invertase, it is hydrolyzes sucrose to glucose and fructose. And it is not involved in high fructose corn syrup. Then the next one. The G protein coupled receptor rhodopsin is composed of a transmembrane apoprotein opsin and 11-cis retinal. A. It is not 11-trans retinal. 11-trans retinal is formed after light activates the L-cis retinal. So it is 11-cis retinal, that is the answer. What is reduction potential? An atom or molecule gain an electron. Answer is option B. +2 question. What is the unit of absorbance which can be derived from Beer-Lambert's law? Absorbance doesn't have a unit. So answer is option D. The radioactive element that is most commonly detected in human body, it is potassium 40. Answer is option A. Then next one. The technique in which separation is based on net charge of the molecule. So based on net charge. Affinity chromatography is based on specific interaction, antibody-antigen interaction, ligand-receptor interaction, that is affinity chromatography. So A is not correct. Ion exchange chromatography, yes, based on cation exchange, anion exchange. So it is based on net charge of the molecule. Isopycnic, it is based on density, and flow cytometry on the base of size, granularity, fluorescence properties, etc. of the cells. So D is also not correct. Then next one. The analytical method that can be used to distinguish flavor compounds. Hydrometry, it measures water content and specific gravity. Then near infrared spectroscopy, it is not used for flavor compounds. Polarimetry, it measures optical rotation typically of sugars. So what is left out? Gas chromatography. So answer is option D. Then next one. Lead levels in drinking water could be determined by AAS, that is atomic absorption spectroscopy. Infrared spectroscopy is not suitable for lead. HPLC for organic compounds, not for metals. GCMS also, it doesn't measure lead levels. So AAS, that is atomic absorption spectroscopy, it is for metals. Then next one. For constructing the glucose biosensor, which of the following is used as a gel? So gel is used as an entrapment matrix to immobilize the glucose oxidase enzyme, which is used in glucose biosensor. So it is polyacrylamide that is used as the matrix or the gel matrix. Okay. Then urea, it is not related to glucose biosensor. Then urease, it is used in urea biosensor. Acrylamide is a monomer for the synthesis of polyacrylamide. So answer here is option D. The next one. Which of the following mechanism of action of polymyxins for inhibiting the growth of the microbes? So polymyxins, they disrupt cytoplasmic membranes. Answer is option B. Then identify the correct statements. Definition of biological species was given by Ernest Mayr. 1 is correct. Photoperiod does not affect reproduction in plants. It influences flowering and seed production. So 2 is not correct. Binomial nomenclature system was forwarded by Whittaker. No. Carl Linnaeus developed binomial nomenclature and Whittaker proposed the 5 kingdom classification. So 2 and 3 are not correct. In unicellular organism, reproduction is synonymous with growth. That is also correct. So 1 and 4 are correct. Answer is C. Then tall plasmid codes for tumor induction of plant crown, that is TI plasmid. Antibiotic methylinomycin biosynthesis, that is also not correct. Root nodulation in leguminous plants, that is by SYM plasmid, SYM. In rhizobia, that is responsible for root nodulation. Then the degradation of toluene. Yes, the tall plasmid is for degradation of toluene. So answer is option D. Which of the following is an example of commensalism? Now commensalism is one organism benefits, the other is neither harmed nor benefited. A virus infecting host and causing disease, it is parasitism. A fungus growing on dead tree. Yes, the fungus is benefited from the dead tree, but the dead tree doesn't have any benefit from the fungus. It is dead. So B is the answer. A bacterium living in the human gut and providing beneficial nutrients, it is symbiosis. A herbivore eating plants for food, that is predation. So B is the answer. Then next one. Name the part of processed antigen that bind to the MHC molecule and recognized by T cells. It is aggritope. Now what is epitope? Epitope is a specific portion of the antigen to which antibodies bind. That is epitope. Immunoglobulin, there are IgG, M, A, E and D. Chaperone, they are involved in protein folding. Now one more I want to tell you what is paratope. Paratope is a part of the antibody that binds to the epitope of the antigen. That is called as paratope. So epitope is for the antigen, portion of the antigen, and paratope is part of the antibody. Remember, Then next one. The chromosomal alteration which causes retinoblastoma, it is deletion in chromosome 13. Deletion in chromosome 11 is Jacobsen syndrome. Translocation between chromosome 9 and 22 Philadelphia chromosome, and it is seen in patients with chronic myeloid leukemia. Then translocation between chromosome 8 and 21. It is most frequent chromosome abnormalities in acute myeloid leukemia. Then next one. Which of the following statement is incorrect about superantigens? Okay. Viral or bacterial proteins? Yes, endogenous in nature. They are not endogenous, they are exogenous in nature. So B is your right answer. Unique binding ability activate a large number of T cells. So that's also correct. Then the first recombinant vaccine produced targeted the surface antigen of hepatitis B virus. Answer is option D. That was the first recombinant vaccine. Then an orphan drug is a very cheap drug. No, it could be expensive because of the development cost. A drug which has no therapeutic use. It has therapeutic use. A drug which act on orphan receptors. No, orphan receptors are proteins that are similar in structure to other known receptors, but their ligand is not yet known. They are orphan receptors. So C is also not correct. So what is an orphan drug? A drug needed for the treatment or prevention of a rare disease. So answer is option D. Then next one. Which of the following is incapable of stimulating an immune response unless they are bound to a larger molecule? It is a haptene. What is ribozyme? An RNA molecule with enzymatic activity, and fusogen, it facilitates cell fusion. Then identify a polysaccharide vaccine. So polysaccharide vaccine, it will have polysaccharides from bacterial cell wall. So here it is HIB. It is Haemophilus influenza type B, and it is an inactivated bacterial vaccine that protects against HIB infection. Hepatitis A is single antigen vaccine, protects against hepatitis A. Salk is inactivated whole virus vaccine. Anti-rabies also inactivated whole virus vaccine. Then next one. The drug which is obtained from leaves. Emintine. It is from the root of Psychotria impecacuna. Digitoxin, it is from foxglove leaves, were used in the arrhythmia treatment. So B is correct. Vinblastine, it is an alkaloid from the leaves of Catranthus roseus, and it is used in the treatment of various cancers. Then tubocurarine, it is obtained from bark of Chondrodendron tomentosum and is used as a muscle relaxant. Then next one. Gray baby syndrome is characterized by circulatory collapse due to excessively high serum levels of chloramphenicol. Answer is option C. Then blood clotting factor rate, it is related to hemophilia A. Creatinine is a waste product elevated in kidney dysfunction. UDP glucuronyl transferase. It is an enzyme involved in chloramphenicol metabolism. So gray baby syndrome is characterized by chloramphenicol. Excessive high serum levels of chloramphenicol. Then next one. In the following diagram, which organ name should be in the place of this question mark over here? So it is small intestine. How through oral administration, It is absorbed by the gastrointestinal tract, then transported to the liver via the blood, from the liver to the bile, and from the bile again it is released into the intestine, then reabsorbed again into the bloodstream through the intestinal walls, and it is returned to the liver. Enterohepatic circulation of drugs. Then what is meant by ADME in pharmacokinetics? It is absorption, distribution, metabolism, excretion. Answer is C. And these four processes, they describe the fate of the drug in the body. Then next one. The term used to signify a preparation that appears identical to the preparation of an active drug but has no biological activity, it is called as placebo. It is identical to the preparation of an active drug but it doesn't have biological activity. So it is called a placebo. Dummy drug, it is an informal term for placebo. It is not a scientific term. Then peptidomimetic, it is a compound mimicking peptide structure and function. Then 3D QSAR, that is quantitative structure activity relation used in drug designing. Yellow region indicate favorable points for smaller groups. Answer is option B. Next one. The functional food which improves gastrointestinal health as well as calcium absorption is fructooligosaccharides. Answer is option C. An induced fit is the process by which a binding site alters shape such that it is ready to accept the drug. The binding site will not alter its shape by seeing that the drug is coming to bind to it. So A is not correct. Binding of a drug to a binding site alters the shape of the binding site. Yes, that's correct. A drug adopts the correct binding conformation before entering a binding site. Okay, it will see the binding site is somewhat triangular in shape. The drug will adopt a triangle shape so that it will bind there. That is not going to happen. So that's not correct. A binding site alters the shape of the drug into the binding. It doesn't alter the shape of the drug. So D is also not correct. The drug which inhibits platelet aggregation by inhibiting the enzyme cyclooxygenase and formation of thromboxane A2. It is aspirin. Abiciximab is monoclonal antibody. It inhibits glycoprotein 2B3A receptor and prevents platelet aggregation. Clopidogrel, again an ADP receptor antagonist and inhibit platelet aggregation. Warfarin, it is an anticoagulant and it inhibits vitamin K dependent clotting factors. Then identify second generation thrombolytic drugs. So second generation thrombolytic drugs, they are fibrin selective drugs and include tissue plasminogen activator and pro-urokinase. And these drugs are used to treat acute myocardial infarction and acute ischemic strokes and acute pulmonary embolism. So alteplase is the answer. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you are new to my channel, if you want still previous year question papers of Kerala SET Biotechnology being discussed, you can refer my channel playlist, that is in your channel name is Omni Bio Entrance. In the playlist you can see for Kerala SET exams, in that you will find. So thank you.